Good morning. So we are starting the Advances in Multi-Omics Approaches to Understanding Eye Diseases session. And I have Professor Cheng and Professor Hai Si as my co-chair. And with that, we start the first talk by Professor Cheng, and that is titled Multi-Omics Mendelian Randomization to Decode Causality in Eye Diseases. Professor Cheng.
Is your name here?
Oh, no. Maybe we can start with the second speaker. Yeah,
Who's that? Do you have your pen drive?
Yes, it is. Yeah.
Played from the pen drive.
Okay. So
Start with the second speaker. My slide.
Yes. Okay. So apologies, we are having some issue with the first speaker slides. So therefore, we are moving to the second speaker, Professor Chakravarti, and he will talk on molecular dissection of primary congenital glaucoma using multi-omics approaches.
So thank you, Kumar, for that introduction, and welcome to this session. So I'd be talking about primary congenital glaucoma and how we have been trying to understand the underlying basis, molecular basis using multi-omics approaches. I don't have any financial disclosures. So primary congenital glaucoma, as we all know, is a developmental disease, and with etiology that involves a restricted ocular outflow because of the development. There is a defect in the trabecular meshwork and the anterior chamber angle causing raise of pressures, and the clinical features would have the HAP striae, blepharospasm, photophobia, and so on. We have been interested in this disease just for the prevalence because it's almost 1 in 3,300 in the state in India where I come from, as opposed to other Western countries where it is not a major problem. It almost resembles something in the Saudi Arabians and the Slovakian gypsies. And the reason we have been interested in is because of the population structure, which practices a lot of consanguinity. There is inbreeding, and that raises the prevalence of autosomal recessive diseases. With linkage analysis, few loci were mapped. The first one, of course, was the GLC3A, which harbors the CYP1B1 gene, then the 3D, which is a bit controversial in terms of classical congenital glaucoma because it has other phenotypes, and the 3E later came up with whole exome sequencings. Myocillin and FOXY1 were also mapped. Myocillin, although mapped for JOAG, was shown to be involved in congenital glaucoma. Later, using whole exome sequencings, there are a few other genes. These are not necessarily loci. The loci are not characterized, but these are genes that were found due to sequencings by whole exomes, targeted sequencing, and so on. If you look into the profile of candidate gene across the globe, CYP1B1 obviously has a major involvement because it has been screened extensively as opposed to LTBP2, TEC, myocillin, and FOXY1, which are considered the 5 major candidate genes. Our strategy has been using multi-omics because using the traditional or the conventional approach, we haven't been able to understand or decipher the entire genetic basis of PCG. So we took a multi-omics approach. One was using the genetic basis of using a combination of whole exome, whole genome, and targeted sequencings, followed by understanding the multi-allelic inheritance and interactions, and thereby undertaking a genotype-phenotype correlations. Using the quantitative traits, again, to understand the genotype-phenotype correlations. And the other was to use proteomics approach using the trabecular meshwork, aqueous humor, and then trying to understand novel proteins that are involved, and then linking all of the 3 together. So earlier we had shown, in collaboration with Dr. Hemant Khanna, who is here, that digenic inheritance, that is inheritance of 2 different alleles of 2 different genes, could cause congenital glaucoma, and TEC was one such which was involved in a digenic scenario. And we also found that depending on where the mutation is, whether it is extracellular or intracellular, they have different phenotypes. We found that these variations also have a corresponding physical interactions. As you see that in wild type, they interact together, the TEC and the CYP1B1, whereas with respect to the mutations, when the mutations were created, there is a perturbation of those interactions, which could be resulting or contributing to the phenotype. On the other hand, if we take only the wild type of one gene, the heterozygous component and wild type component of one of the genes, they do not show such interactions. We also showed that since angiopoietin, which is a ligand of TEC, that also has different kinds of behavior in the HUVEC cell lines, and this has already been published. Furthermore, we showed other genes like the serine protease that also interacts with CYP1B1, and knocking down of the serine protease in mice resulted in hypoplastic trabecular meshwork. Similarly with GLIS, which also interacts with other glaucomatous genes like FOXC1, and again, knocking down GLIS results in hypoplastic trabecular meshwork, and so on and so forth. Recently, we also found through our studies on whole exome sequencing that cilia genes, particularly CYP164 and INPP5E, could also be involved in PCG pathogenesis, and they also interact amongst each. There is a physical interaction between CYP and CYP1B1. Taking all of this, we also tried to identify that can we have different measures of genotype-phenotype correlations, and we tried to take quantitative traits not only with respect to the corneal diameter, intraocular pressure, etc., but we looked into the preservation of the retinal ganglion cells and tried to do photopic negative response on these childs. And we found that given the signature of multi-allelic inheritance, there is a difference in the photopic negative response in the children who develop congenital glaucoma as opposed to those who do not. And we also looked into the correlations of those parameters, the clinical parameters with parameters of the PHNR, and we find that individuals or patients who have multi-allelic changes, they have a stronger correlation as opposed to those on your right-hand panel who do not harbor multi-allelic interactions. So in short, candidate genes have 60% unknown. When we use other forms of sequencing, deep sequencing, like using a targeted panel, we reduce that to another 48%. Using whole exome sequencing, we reduce that further, and so on and so forth. But what is interesting is that 20% or so are the co-occurring alleles, as I showed, where they do have corresponding genetic as well as physical interactions. And this is not necessarily the case with congenital glaucoma. Even with very large cohorts of retinal dystrophies, there was a study by Steve Diger and others who showed that even using whole exomes, whole genome, and using copy number changes, they were also having another 30% of the cases which were not accounted for. And so the cases that are not accounted for in our cohorts could also be because there are mitochondrial changes, which we are looking into now. There could be copy number variations, epigenetic changes, which have not yet been characterized. Interestingly, these co-occurring variants, if we look into the whole repertoire of genes, they have multi-allelic inheritance. The red boxes indicate that they are homozygous, blue, they are heterozygous, and yellow are compound heterozygous. So on the z-axis, you have the cases, the y-axis has the genes, and this is just a snapshot to show that all the patients have different forms of multi-allelic interactions. Interestingly, this kind of phenomena is also seen in controls, and controls also have these kind of signatures. And so while determining what actually is the actual signature for the phenotype, we need to kind of eliminate these kind of combinations. Added to that, India presents a lot of ethnic diversity, and that actually increases the problem because many of the data that you find from India are based on the controls which are based on 1,000 genomes or the GenomeAD database. But India never participated in those. So we have surrogate populations in those, like for instance, the Punjabis in Lahore, Gujaratis of Houston, Telugus of UK, Sri Lankan Tamils, and Bengalis of Bangladesh. We generated our own controls from a longitudinal cohort, which we have been following up for the last 20 years. And just for the set of glaucoma-associated genes, we found, and all anterior segment genes together, and ciliary genes together, we found that there are 390 or close to 400 pathogenic variants, of which another half of them are present in the controls. Now, what happens is when we overlay this data on the 1,000 genome or others, you find that in the 1,000 genomes, the allelic diversity is much less compared to what you find in the local population. So that's, again, a kind of a red flag that when you consider data from India, you need to ask for the local cohort, and they will have more of allelic diversity, which will render that some of these variants, which are found on the background of 1,000 genomes and nomads, to be false positives. We again, then we looked into the expressions of these genes, and we found that anterior segment genes could be involved in the development of anterior chamber based on their levels of expressions. We also found that these are not present in the adults. These are more expressed in the childhood cases. And when we do these interactions, we find, to cut a long story short, that there are two different pathways that stands out, the hedgehog signaling and the Wnt signaling pathway. And these could be involved in congenital glaucoma. We then took into proteomics, we took cases which are, based on the genetics, we categorized the cases into homozygous, heterozygous, and those without variations in CYP1B1. And we found that there are certain unique proteins which are present in the trabecular meshwork of these patients. And what we found is that homozygotes have an overexpression of TGF-beta, and those without variations, which are PCG, but no variations in CYP1B1, have a higher expression of CYP1B1. These unique proteins, the TGF-beta, are upregulated or enriched in homozygous cases as opposed to those who are heterozygous. And we find a different kind of a pathway because the TGF-beta is regulated by endothelin-1, which is a vasoconstrictor. And what we find is that in cases which do not harbor CYP1B1, we have overexpression of CYP1B1. Those with homozygous mutations usually do not have CYP1B1, and endothelin-1 is overexpressed in the aqueous humor of these patients who harbor CYP1B1 variations, indicating that the pathogenesis could be due to the ECM dysregulation, and which is contributed further by other ECM matrix and the endothelin-1, which actually increases the TGF-beta and reduces CYP1B1. So this is the working hypothesis that we have currently. One, which is through the hedgehog signaling, one through the Wnt, the different interactions of the genes, and there is a cellular death, cell death component also, which is due to the reduction of CYP1B1, overexpression of endothelin-1, leading to TGF-beta. So to conclude, it is a complex disease, which was earlier not thought of as a complex disease, and it has multiple variants with varying magnitudes of effect. So we need to consider these multi-omics approaches, and combinations of sequencing strategies actually revealed signatures, which we have validated some of them, not all of them. And that may reveal ocular angle development and raised IOPs, which are again complex mechanisms, and it could be deciphered through multi-omics approaches. And the functional interactions of genes in the maintenance of the structures may hold key towards understanding of disease pathogenesis. I'd like to acknowledge the patients, the volunteers, the funding agencies, the collaborators. Then thank you.
Thank you, Shubho. We can go for one or two quick questions. If not, yeah.
Just ask a
Yeah,
question.
go ahead. Yeah.
I was just noticing the great work you've done with proteomics, like from trabecular measurement. That's impressive. I'm not aware of many people who've done that. Do you see any correlation between the protein levels that you measure in the eye with sort of like systemic protein measurements? Like how much you actually measure systemic? Do you see traces of those signals like peripherally?
No, what we saw is with regards to, the proteins, what we had extracted We had loaded almost equal amount, both in cases who harbored mutations in CYP1B1 because that was the categorization, and those who did not have. So these did not have were the ones who would be considered as normals, right? So the proteins were, the protein amount was the same. Signatures were different because if you see in that pie chart, I mean, I rushed through it, there were certain unique signatures which were specific to those cases who harbored mutations, those who did not harbor. Yeah, but there were majority of the, I think 2084 proteins were common to them, but the number which will be unique would be 100 or so among the homozygous cases, and those without mutation would be around 40 or so.
I was
Yeah.
thinking about the potential to actually use those biomarkers.
Right. Yeah.
In the diagram where you showed that you were looking at genes other than CYP1B1 as well, and you showed that there are patients in which there's CYP1B1, and along with that there'll be some anterior segment gene like
Correct.
a PITX, or were their phenotypes distinct in any manner, or apparently they were the same?
No, these were cases who were classical cases of PCG.
Okay.
So during our inclusion itself, those who had a variable phenotype were not considered. So within the classical cases of phenotype, we wanted to see what is the extent of the genetic diversity that we are able to see.
And were those variants benign or SNPs, or you only marked them because they had some sort of a higher pathogenicity?
These
or
are all
level?
the rare alleles which are pathogenic
Okay,
with a Revel score of 0.5 and above.
Above.
Yeah, it was there somewhere in the
Okay,
bottom. Yeah.
Thank you.
Yeah.
Thank you, Doctor.
Sure. Thank you very much.
And now it's my pleasure to introduce Dr. Qingyu Chang from Singapore, who's going to talk about multi-omics Mendelian randomization to decode causality in eye diseases.
Good morning, everyone. First, I want to thank Professor Penn and scientific committees for their invitations, and allow me to share with you my recent research work in Singapore. So I'm going to talk about the Mendelian randomizations. So if we want to study the association between the risk factor and the disease outcome, using a traditional epidemiology approach, right, there are always multiple confounders, right, between the risk factor and the disease, right? For example, in this case, I mean, the confounder can be either unknown confounder or what we haven't measured. So ideally, So basically, so if, for example, if we observe association between a risk factor and disease, it doesn't imply causation, right? Because basically, this association was confounded by many other factors. Now, let me give you an example. If you want to study the effect of nutrition on myopia, right, there's always other confounders such as outdoor activity, sunlight exposure, and so on. So how do we design a study which can tease out the causation between the nutrition and myopias? So for example, ideally we want to assess the effect of omega-3 on myopias. We should conduct a randomized control trial, RCT. However, this RCT is not so feasible due to a few challenges. For example, there's always a relative long time to assess the treatment effect, right? And second, we also don't know what's the optimal dosage from the omega-3s. And the adherence to the intervention, in particular in children group, is also a challenge. So, but we know that, we all know about Mendel's second law of inheritance. So according to this law, two alleles from the same genes will assert independently of allele from different genes. So that is allele that is transmitted from parent to offspring is randomly determined. And this is actually equivalent to randomized control trials. And so you can see that because of this randomization natures, right? So actually each of the two alleles in a parent has an equal chance of being transmitted to the offspring. So in the RCT, the patients are randomized into either intervention group or the control group. then we can compare the treatment outcome between the two groups. Whereas in a randomized randomization, AIOS are randomized to two groups, and we can compare the outcome between the two groups to see whether it's due to the effect from AIOS. So in MR studies, right, we use genetic variant as a proxy for the treatment effect. So we hypothesize that people who has the omega-3 rising AIOS will have a similar effect as taking the omega-3 supplement in the RCT. So both of them lead to the higher blood level of omega-3, and that might modify the risk of myopia. So this MR design allows us to determine the causal effect between omega-3 and myopia. So in a randomized RCT, so if we want to estimate the effect of treatment X on outcome Y, actually we can use instrumental variables. And in this case, in the RCT, the instrumental variable is the assignment in the RCT setting. So basically, with the instrumental variables, we can the effect of S on Y can be estimated by the GY association divided by the GS association. So by doing this, we can get an unbiased estimate of the treatment effect. So when we apply this idea in the context of genetic analysis, right, we can replace the assignment with a genotype, and we can also replace the treatment with the so-called intermediate trait between the genotype and outcomes. And this is so-called the Mendelian randomization analysis. So now if we want to look at the causal association between omega-3 and myopia, what we need to do is just get a list of genetic SNP that is associated with omega-3, and also a SNP that is associated with the myopia. So by using this equation shown here, we can estimate an unbiased effect between the omega-3 and myopia. So basically in this work, like, we include the GWAS result from 18 fatty acids from the UK Biobank. And the fatty acids are measured by the target MMR approach. And this omega-3 basically is a subgroup under the polyunsaturated fatty acid. And we select 5 MR model to address the potential polytrophy effect and other analysis assumptions in the MR analysis frameworks to ensure that our results are robust. So we use about 20 to 15 SNP for the analysis. And this is a number of SNP we use for each of the fatty acids. And this is the proportion explained by the instrumental variable in each of the fatty acids. So the slide shows the fatty acids are associated with a more positive 3-core equivalent. That is the low risk of myopia. So those fatty acids are the omega-3 to total fatty acid ratio, DHA, and so on. And on the other hand, this slide shows you the fatty acid, that is all fatty acid associated with higher risk of myopia, which include the omega-6 and omega-3 ratios. So basically, in summary, MR analysis suggests that higher omega-3 and HDL level are protected against myopia. And this is of public health significance because taking omega-3 supplement actually is potentially a very feasible approach for the children group. So likewise, we also use a similar approach and to study whether there is any causal relationship between omega-3 and AMD. We also found that the higher plasma level of omega-3 and DHL level are protected against AMDs. And in this work, we look at the association between the metabolomics and open-angle glaucoma. And we identify the HDL3 cholesterol is significantly associated with open-angle glaucomas. But we cannot find another cohort for the application purpose. So we use this MR approach to validate our finding. And the MR analysis again supporting that HDL3 levels is causally associated with open-angle glaucomas. So the second application of multi-omic MR analysis is the drug target MR approach. So basically in this approach, right, we selected genetic variants affecting gene level, gene expression level or protein level to simulate the drug intervention effect. So typically, genetic variants located in the cis region are chosen as instrumental variables. For example, we can include a SNP plus minus 100 kb within the gene boundary for analysis. But before you do analysis, we also do some quality control to make sure that we can get a reliable result. So let's take a reference from this paper published in Nature Human Genetic Behaviors. So basically in this work, the author integrated protein and gene expression data and conducted a drug-based MR approach. So basically in this work, they identify 3 proteins that affect the gene expression level in the brain and suggestion that these 3 proteins are the potential drug target to change the alcohol drinking behaviors. So in this work, basically the author also used a similar approach, and they identify the A protein level in our plasma, which might affect the risk of POAG. And they also suggest these 8 proteins are the potential drug target for open-go glaucomas. And the third application of multi-omic MR analysis is the mediation analysis, particularly when leveraging multi-omic dataset to tease out the direct and indirect effect. So this is quite straightforward because using this MR analysis design, actually we can just estimate the indirect effect and direct effect, and the mediated portion just the indirect effect divided by the total effect. So using this approach, right, for example, in this paper, they look at the association between the gut microbiome and the risk of AMD. So basically, they found that, for example, this bacteria, Schleichia bacteria, can reduce the risk of dry AMD. And this was through the higher level of glutamine. And glutamine mediate the 70% of the association between this bacteria and dry AMDs. And just give you another example of the mediation analysis. Basically, we know that in high myopia cases, it's very difficult to diagnose glaucomas. So we also did an MR analysis to tease out whether there is a causal effect between myopia and open-go glaucomas. So we showed that higher degree of myopia can causally link to open-go glaucomas. And we also did the MR analysis based on between the bidirectional causal association between IOP and myopias. And through the mediation analysis, we also showed that IOP had the most significant mediation effect on the association between myopia and open-go glaucomas. And the effect is much stronger than other variables such as nerve fiber thickness and the cup area and disc area and so on. So basically, this suggests that even we are not sure that whether a myopia patient has glaucoma or not, or no matter they have a glaucoma suspect or not. Actually, if we provide them with IOP-lowering medication, for long term, it still can reduce the risk of open-go glaucoma. And this is all I have. Thank you very much.
If there's any questions from the floor.
Thank you, Chenyu. That's always interesting. It was very clearly explained, the Mendelian randomization approaches. I'm wondering, from your perspective now, having really become an expert in this area, what you would recommend as kind of quality control or expectations from these kinds of experiments. So, I kind of have 2 general questions. One, what sort of effect size would you consider significant? And is that really related to the p-value? And what kind of cutoff, is a p-value of 0.05 actually significant in this setting? Do you need to correct for multiple comparisons for the number of SNP, vehicles that you're using? Are the SNPs that you're using, if you're using more than one SNP, are they all correlated or not correlated? Do you account for differences if they're correlated or not correlated? It's kind of a murky area right now in terms of deciding if something is significant or not in this kind of approach. I'm just curious what you might say to that.
Thank you, Jenny. I think it's a very good question. Actually, we see many more paper on the MR now compared to 5 years ago, right? Basically, because to do MR analysis, basically you don't need to collect your own data, right? You can just, download the GWAS summary statistic from the open source, and you can do a MR analysis on everything. But it become too much, right? So my current approach is that I use MR analysis as a validation tools to validate the association study from epidemiologists or from clinical observations. So by doing this, the question we want to address, for example, the association between myopia and glaucoma, right? This is our clinical question. But we can use multiple approach to address, to validate these associations. So I'm not suggesting we use MR for everything, right? You have to ask clinical meaningful questions before you want to do an MR. And my second comment is about what's the P cutoff we should use for the analysis. Basically, there's no consensus on this, right? So what I use that, for example, why there are so many different MR models, right? Because each of them have its limitations. Because each of them only address maybe just one of assumption, under multiple, one of assumption of the amount of multiple assumption under MR analysis. So what the approach I use that, for example, if you use five different MR model, you should maybe set a more stringent criteria, right? At least 3 of the 5 models showing significant P less than 0.05 and also the same durations, then we declare it as significance. But again, there's no consensus what is better way to do this. I hope I answered your question.
And we may just have time for two very quick questions from Dr. Akiyama and Dr. Chakrabarty.
Okay. Thank you for the great talk. You showed the association, causal association of HDL3?
HDL3
on POAG.
Yes.
In the GWAS AMD, so we found many loss associated with repeat, but so we have written know about genetic association of genetic loci associated with repeat on the risk of POAG. So do you have any idea on it?
No, actually, we can discuss more about this. I didn't look into this, but it's interesting observations here.
Okay, thank you.
And a very quick one, Dr. Chakrabarty.
Question, like for instance, in MR, when you're looking mostly into the causality, and these are largely based on cross-sectional data, right? You are taking GWAS data and you are taking the phenotype data at that point of time. But these are actually progressive data, like when you talk about myopia, when you talk about glaucoma. So how are these adjustments done at a later time? Because the cross-sectional, actually, it should have been longitudinal, but in cross-sectional also you can predict a longitudinal outcome.
But basically this is also the same issue with all the GWAS analysis, right? Because our phenotype change over time, but our genotype is fixed at birth. Right, so even it's cross-sectional data, basically, when you look at genetic association, basically, it's more like the perspective, right? Because the genetic, the risk factor already fixed at birth, right? They won't change over time.
No, the germline changes are fine. I'm talking about the clinical data, which is progressive. Because there also, let us say if we take GWAS data, we're taking only at that point of time when the individual has been sampled, whether he had the phenotype, milder phenotype, moderate phenotype, like. But over a period of time, that phenotype can change.
Yes,
so in MR, how do we adjust for the longitudinal outcome?
Well, this is an interesting comment. Actually, we haven't looked into this yet, but I believe this will require some new method for this type of MR analysis. Yeah.
Thank you so much, Dr. Chin Yee.
multi-omics and MR looks like it's time for retirement, actually. Okay, we move on to the next speaker, Professor Heisey. And he's going to talk on multi-omics analysis of regulatory networks influencing intraocular pressure and glaucoma. Professor Heisey.
Thank you so much, Dr. Kumar. Great, thanks. Just before I get started, just wanted to thank the organizers for having me at another amazing APO session here in India. Great to be here. And I have no disclosures to make. So quite a lot of what we know about elevated IOP and glaucoma comes from multi-omic studies or like single-omic studies. So we know there's like hundreds of genes that are involved in both elevated IOP and POAG, and they are expressed in a highly tissue-specific manner. We know that age-related driven epigenetic status changes around those genes can at least in part influence and dictate the late onset that we see for a lot of glaucoma cases in the population. And we know that metabolism, specifically photooxidative stress and lipid metabolism and others are important to the homeostasis in the anterior segment and trabecular meshwork. Now, talking about multi-omics, like often the base layer of the omic stacking are the GWAS or the genome-wide association studies. There are several of them that have been published over the years. The most recent one is something we are still working on, and we put together a cohort of 350,000 individuals, more or less, with both IOP measurements and SNP data. And as usual, the more power you have, the more you know about the genetics of a disorder. So we've identified a couple of dozen actually new genetic regions that are annotated through those like vertically oriented labels in that figure. We also see like higher levels of association, but these are known loci, some of which are actually still shown on this figure. Now, that sort of genetic knowledge is useful to make predictions. And although imperfect, our genetic predictions are getting better, and we have this unprecedented ability, and still it's work in progress, but to identify individuals who are going on to become glaucoma patients or who'll stay controls for the rest of their lives. What does that tell us about the gene functionality? Something but a little. So we know, for example, there are a few recurring themes among the GWAS association studies. We know, for example, just to cite like a few random annotation labels, that angiogenesis, cell adhesion, extracellular matrix, lipid metabolism, and much more are overrepresented among the top GWAS hits. But we know very little about how those genes link together in a coherent cascade to actually elevate IOP and cause myopia, or conversely to contribute to good health in the eye. And this is not just genetics data. Like every single large omics biological data analysis has the same limitation. So these findings tend to be associative in nature, so they tell us little about causality. They are annotation or enrichment based, and they just heavily rely on co-occurrence and co-localization with little thought about causality. And these are all good questions that integrated omics approaches can answer. And specifically here, I'm just going to try to focus on the obvious ones, like how are the genes and proteins working together to maintain health or to increase glaucoma risk? And are there any specific biological cascades that contribute to those conditions? And again, the implicit question is if they can be exploited for new therapies or otherwise in the clinic. Just a sort of like so essentially just to bring you back to like biology 101 on transcription. We just heard from the excellent talk that Dr. Chakrabarty gave that there are so many genes harboring mutations that can cause very dramatic, life-changing, early onset and clinically severe glaucoma in early life. And those genes tend to be in the protein coding parts of the gene, which are shown here, like using the red symbols at the bottom. But the SNPs that we identified through genome-wide association studies tend to cluster at the other part of the gene earlier on in the sequence, around the enhancer and the promoter region. So about half of the SNPs that we've identified for our GWAS of intraocular pressure and glaucoma are actually in that region, and they have significant effects over the level of the mRNA that is being transcribed, adjacent to those like sites of association. Now, the same gene, we see an overlap, so the same genes can harbor mutations, the same genes can actually have EQTL effects associated with glaucoma and IOP, and here I'm just bringing a few examples.
So TAC genes has important SNPs that are common in the population that are associated with changes in the transcription process, and so is LMX1B, the gene behind nail patella syndrome, and the ABO SNPs apparently are associated with both IOP and have EQTL effects. So this is all stuff we knew. Like, how about, so the real question here is, like, is there any remote ability from those genes? So can they actually change something remotely elsewhere in the genome, something that they don't do physically, but they do through interactions, like protein-protein interactions, somewhere, if they can actually leverage any biological cascade to change something down the stream. To do this, to answer this question, we actually assembled a large data set. Here, just reading like mistakenly, no, it's not mistaken. So here we just like assembled this large data set, like thousands of individuals with both peripheral plasma protein measurements and SNP data. And we found that not just in the context of IOP and glaucoma, but some of the major hubs of interaction, trans interaction, like distant interactions, were just revolving around the ABO gene that we just mentioned is important for the development of like elevated IOP and glaucoma. So the ABO, like the blood group locus, had some really outrageous like levels of association and all over the genome. More in the context of a glaucoma slash like elevated IOP, the locus at the ataxin-2 and SH2B3 genes that are very like next to each other on chromosome 12 is also very important. Of all the genes that participate in mechanism we believe that are associated with IOP and glaucoma, those two genes are responsible for 99% give and take of the interactions that we see. And this is a simplified version of the same chart. Essentially, every arrow is in a significant association between a SNP and a protein. And the genomic loci where those SNPs are located are actually shown like in faint blue. And the genes that encode the proteins that those SNPs are associated with are actually highlighted in yellow. Unfortunately, those arrows have slid a little. But again, a vast majority of those interactions still revolve around ABO and the locus on chromosome 12. There are a few limitations to what I just mentioned. So this is like an oversimplistic representation of the reality. So those arrows often involve like tens, if not like hundreds of other proteins along the way. And blood proteomics can be a blunt tool occasionally. There's good reason to use it, but there are limitations. Typically, when you do plasma proteomics, you only focus on very abundant proteins that you can see, but that kind of excludes like transcription factors, which are important in their regulation. And in the blood, you actually measure pretty much what's washed in the plasma, but you don't have the tissue specificity you may need. Therefore, transcriptomics can actually be a more sort of delicate and elegant tool that we decided to use as a next step. And I'm really grateful to Dr. Chingyi for explaining the principles of the Mendelian randomization in a much better way than I would have time to. But basically, the method involves like instruments, like SNPs are used as instruments. They have a certain effect over an intermediate parameter of some sort. And then we are interested in understanding better the relationship between that intermediate parameter and outcome of interest. Now, again, like those intermediate parameters and outcomes of interest can be anything we want, but in this context, I'm going to talk about both the intermediate parameter and the outcome being levels of transcribed mRNAs. And I knew that it would be like time would be like very short, but to do like using those so simplified Mendelian randomization to mass scale and using some pretty stringent false discovery rates, we just discovered quite a few relationships that we believe are actually quite convincing. And here I'm just like showing like a part of the picture that is like slowly emerging, and we actually see even at a transcription whole blood transcriptomics level that the ABO locus has the ability to regulate the TEC peripheral expression in the blood. In some tissues, also like myocillin, somehow in the blood, but a lot stronger in other tissues that were available to us. They don't seem to be immediately related to glaucoma, but we saw very strong regulatory effects in skin. And those interactions, again, like those arrows, in a way, like hide the true complexity of those interactions. So they are an oversimplified way to represent what we believe to be the reality. So the takeaway sort of message is that the ABO have very significant trans EQTL and PQTL effects that were some of the strongest that were ever detected on humans. The ABO locus may be a regulator of pathways that are important in angiogenesis, both systemically and in glaucoma genesis narrow context. The ABO blood group B, or like SNPs that are in strong LD with the ABO blood group B, tend to elevate the TEC expression in peripheral blood and other tissues. And they have like remote regulatory effects over other glaucoma genes. Attacks into alleles, the attacks into alleles that tend to be associated with increased levels of IOP tend to decrease attacks into expression, and they tend to increase the myocilin gene expression. And this is again like a partial view of what is slowly emerging. So many funders, like collaborators, institutions I wanted to thank, and so many of you are actually in this room, so thanks for your support. But this photograph, again, is a lot of people who are just doing the IgGC genetic analysis, and I'm grateful to all of them. Thank you so much again for your time.
Thank you, Professor Heisey. So questions, comments, please. Yes, sure.
Circulating plasma, because you're looking at a very focused disease. So,
yeah.
Because again, as you would know, the abandoned proteins would be much higher there, like albumins and other things would go up. But so the second related question was the ones that are surrounding the ABO locus, were they the low abundant proteins that you got?
The protein, plasma proteins, yeah. They were like low abundance. They were high abundant. In the plasma, we only measure those ones that are above a certain threshold.
Right. So the first question was the rationale of using plasma,
right.
Circulating plasma.
So two like very basic sort of reasons. Like first of all, that is like availability. It's cheaper, it's easier, it's more practical to actually do it on plasma. And there's like a hidden sort of like a purpose, if you will. Identifying biomarkers. Say that you identify this biomarker that diagnoses glaucoma like at birth or anything. It's not going to be like practical to actually get like an eye biopsy at birth or anything. So you actually kind of ideally want to do that in plasma. So that's kind of like a hidden wish we had.
Thank you. So, Jenny.
Piero, thank you for that really super interesting talk.
Thanks.
I have a question more around methodology. And that is, in your initial analysis where you looked at all of these, protein interactions and kind of funneling in on these two hubs of Ataxin-2 and ABO, how do you account for, or how do you correct for, or how do you remove the bias of proteins that have a lot of protein interactions? So, for example, Ataxin-2 does a lot of stuff in the cell, and so just sort of naturally, if you don't correct for that in some way, those are going to be identified as important hubs because they're pleiotropic. They just do so many different things. Where there could be a very specific interaction, just like one interaction between two proteins that's critical to glaucoma, but wouldn't have survived your analysis because it wouldn't have formed those two hubs.
Again, it's going to be like the first part of the quick answer is going to be like how we define those. What is an interaction? Again, like to me, this is actually stuff that we are exactly looking for. Essentially, what Ataxin or ABO can actually do downstream, like what those SNPs will never interact directly with the transcripts, like somewhere like in a different chromosome. But it's essentially through the Ataxin protein doing something else elsewhere in the body that we're getting those associations. So essentially, that was kind of like the purpose why we did that as opposed to like a byproduct. But again, like think about bias. Bias would be like population stratification. Again, like blood group is we had to, part of the reason why we're doing Mendelian randomization, we're doing that at a much more sort of like careful way than I showed here, is that again like that could be like a huge source of bias. But I'm not very worried about the multiple sort of pleiotropic effect Ataxin has, because essentially there has to be a cascade underneath that, and that cascade is essentially something that we want to characterize.
So I have a question, Professor Heisey. So if I understand it correctly, what you're suggesting is that the blood group is a platform, and then if you have B blood group with TEC, then you get glaucoma, right?
You're more predisposed in average to have glaucoma.
Okay. So my question is, are there any other eye disease or human disease which has such kind of a link?
Lots. Again, like lots. Again, like gastrointestinal cancers are related to blood ABO blood groups. Like
stomach.
lots of them.
Yeah,
we
that's
see,
a known, very long known
everywhere.
fact,
Like
yes.
a ton of immune responses. Now, again, I specifically focused. It's a much longer story, but very briefly here, I just focused on that because that's a massive effect. What we are seeing actually, those SNPs that we use for as instruments and those effects that we see, they actually change from tissue to tissue. So we actually see, like a taxane interaction, we see the same taxane effects over somewhere else, but actually the genes, the SNPs that we use as instruments are slightly different. So they're actually located somewhere else in that promoter. So that shows like a certain tissue-specific activity that we found very interesting, but I didn't have time to show here in full.
Okay. Very interesting. Thank you, Professor
Thank you.
Heisey.
Thank you, Peter. So may I introduce the next speaker, Dr. Masato Yakiyama from Japan, and he's going to talk about genome-first integrated omic approach to IDEs.
Okay. Thank you for the introduction. My name is Masato Akiyama from Kyushu University, Japan. So first of all, I'd like to express my gratitude to all the staffs and organizers of this conference. So I'm going to talk about genome-first integrated omics approach to complex eye diseases. My financial disclosure is shown on the slide. So today I'm going to talking about two topics shown on the slide. The first is genome-first integrated omics approach, and next I would like to discuss and review future directions and potential applications. Okay, multi-omics analysis can be generally classified into two types: a phenotype-first approach and genome-first approach. In the phenotype-first approach, we compare the molecules such as DNA, RNA, metabolites, and proteins between individuals with and without diseases. And we can identify differently expressed gene or proteins. So in contrast, genome-first approach, we generally utilize the GWAS analysis, the result of genomic analysis, and integrate it with other omics data to interpret it biological findings. So let me explain why I focus on genome-first approach. So as you know, according to the central dogma, our molecule in our body are synthesized based on DNA sequence. So we can find obvious difference in tissue and organs in the affected individuals compared to healthy ones, but we cannot recognize the molecular mechanisms. So on the other hand, we can determine differential expressed genes or protein by performing transcriptome analysis or proteome, but we cannot assess its causality because of reverse causation. So on the other hand, germline DNA sequences are not changed by acquired factors such as diseases and environmental factors. So genome-first approach can identify causative molecules and biological mechanisms independent of reverse causation. So as you know, so many eye diseases are influenced by genetic and environmental factors. In case susceptibility is influenced by both genetic and environmental factors, such diseases are called complex diseases. So genome-wide association study is a method to identify genetic loci associated with human phenotypes. As shown on the slide, this analytic method has been applied to multiple eye diseases and identified many genetic loci associated with diseases. However, GWAS has obvious limitation. So I'd like to illustrate the limitation using this slide. So we performed, in recent study, we conducted a genome-wide association study of age-related macular degeneration in Japanese population and identified two new loci. And one of the regional association plot of the identified loci is shown on the right. In this identified locus contains, this locus contains multiple positional candidate genes. And we cannot determine which genes is responsible for disease onset. So integration with other omics data is essential to narrow down the candidate gene. As you know, omics is a comprehensive study of molecules such as DNA, RNA, metabolites, and proteins. And it is also possible by integrating with DNA data, we can identify genetic locus associated with molecular levels. Genetic locus associated with molecular level is called quantitative trait locus. So if the association signals between GWAS of diseases and QTLs are similar, we can recognize the molecule is associated with disease, with causality. So in this slide, I illustrated the analytic procedure of multi-omics analysis in each. First, phenotype first approach, we evaluated and quantified molecular levels and compared between individuals with disease or without disease. And we can identify genes differentially expressed, gene or proteins differentially expressed, but we cannot assess its causality. So on the other hand, our genome first approach utilizes the summary statistics data of GWAS and integrate that with other omics data, and so we can identify causative genes or molecules. I also would like to note that we can integrate GWAS results with other GWAS results. So we perform by utilizing the association of GWAS results in AMD GWAS, we compare the signals of association between age-related macular degeneration and central serous chorioretinopathy, and found two loci showing similar association signals between these two diseases. This observation indicates that these two diseases share the same biological mechanism of disease onset between these two diseases. Lastly, I'd like to discuss future directions and potential applications. Recent single-cell analysis technology enables evaluation of various omics, including RNA expression, protein levels, chromatin accessibility, and histomodifications. So over the past years, single-cell analysis have been applied to various eye tissues. Here I summarized the studies conducted single-cell analysis across different regions of the eye. So as you can see, a single-cell RNA sequence has been extensively investigated. In contrast, epigenetic states, including chromatin accessibility, have been evaluated only in lens and retina. So therefore, I anticipate that further investigation of epigenetic states and integration with genomic data will provide insight into the pathogenesis of a broad spectrum of eye diseases. So also, recent advances of single-cell analysis can evaluate the expression level while preserving spatial information. So integrating GWAS with this kind of result will further expand our knowledge on ocular eye diseases. Also, I would also like to introduce a new proteome method, OLINK. So this method allows quantification of proteins in a small amount of biological resources, such as tear fluid. Then conducting QTL analysis and integration with GWAS results will further expand our understanding of eye diseases. So now we are conducting a proteome analysis of tear fluids in patients with IgG4-related disease or thyroid eye diseases. Okay, so I'd like to summarize my presentation. So integration of genomic data with multi-omics resources can pinpoint genes associated with diseases. Therefore, genome-first omics analysis can reveal causative disease mechanisms. So also, I mentioned that single-cell omics analysis and proteome in small amount of liquid material will provide novel pathogenesis of eye diseases. Okay, so I'd like to mention my collaborator, and I'd like to show my appreciation. And this is the last slide. Thank you so much for your attention. And I'd like to note that we are going to organize the 130th annual meeting of the Japanese Ophthalmological Society. And if you have a chance, please join us. Thank you so much.
Any question from the floor? So I have one question, sir. So we always like to integrate multi-omic data set, right? But we know that compared to other diseases like cancer, right? For eye disease, we don't have so many tissues allow us to do the gene expression level or protein level. So what's your solution to these challenges?
Yeah, thank you for raising an important point. I agree with your idea, and it's very difficult to investigate the expression level or protein level of eye tissue. So I'm expected that using proteome in small amount of liquids, such as tear or anterior chamber humor or vitreous body, may help to solve this issue.
Any other comment, question? If not, thanks.
Thank you.
Yeah, thank you. Thank you much. Yeah.
Great. So it's my pleasure to introduce the next speaker. Professor Janie Wiggs from the U.S. is going to talk to us about new insights into disease mechanisms from glaucoma genetics.
Okay, great. Thank you so much. And thank you to the organizers. I first came to India 20 years ago, where I first met Kumar. And it's really been a pleasure. I've been here multiple times since then, but it's really been a pleasure to see how India has changed, the progress that's been made, and have something as beautiful as this convention center is really quite interesting. Okay, I'm going to talk today a little bit about some insights that we're gaining into disease mechanisms based on genomic studies for mainly primary open-angle glaucoma. These are disclosures, none of which are relevant for today. So I think you all know, and I'm not going to spend a lot of time here, but glaucoma is an important cause of blindness throughout the world. And importantly, early detection and treatment are really necessary to preserve sight in affected people. But disease screening and risk stratification are challenging. It's hard to identify people who are truly at risk for disease before they develop damage to their optic nerve. And as a result, many affected individuals are undiagnosed. So that's really the premise for why many of us in this room have been engaged with studying genetics and glaucoma for many years now. Glaucoma turns out to be one of the most heritable of all human diseases, and we've had a lot of success in GWAS and other kinds of genetic analyses. There are now more than 300 genes contributing to disease risk that we've identified from these kinds of studies. And so I present a little diagram here as we're beginning to understand pathways and processes that these genes fall into. We are beginning to identify some of the mechanisms that could be underlying disease in glaucoma that's really being illuminated by these genetic studies. So today I'm going to give you 3 examples. I can't obviously talk about all 300 of these genes or even all of these different processes that have been identified through our understanding of what these genes do, but I want to give you 3 examples just to shed some light on kind of where we are in terms of our investigations and what the future could look like in terms of understanding the landscape. So the first one I want to talk about is mitochondria. You all know that mitochondria is the powerhouse of the cell. It's the source of ATP for cells. It keeps cells alive. Mitochondria is particularly important in glaucoma because the retinal ganglion cells that are really the target of the disease process have a high energy requirement and are very dependent on mitochondrial function. So one of the things that's emerging is that the mitochondrial electron transport chain, which is necessary for ATP production, the first complex of that chain, complex 1, has 45 different subunits. So it's a very complex subunit. But 2 of these subunits have now been implicated in glaucoma. I'm going to give you a little bit of data on each of those. So the first is NDUF-S3, which is an important important protein that forms part of the complex. This is a nuclear encoded gene that then is transported to the mitochondria to function. And this gene has been associated now with POAG in gene-based analyses from the last GWAS that's been published, Han et al. In Nature Genetics, with a very significant p-value. So this indicates that dysfunction of complex 1 leads to mitochondrial dysfunction and potentially then increases susceptibility to glaucoma. The second protein subunit of complex 1 that's interesting is also a gene that's nuclear encoded, FASKD1. And here, this data comes from an exome-wide study that we carried out, and we found 2 independent rare missense alleles in this gene, both showing nominal association, but significant effect sizes. And this is a protein that regulates a subunit of complex 1. Decreased FASTK1 leads to increased mitochondrial ND3, which is one of the subunits, and increased activity. And this has come from some functional studies carried out in HEK cells using CRISPR. And I also just showed you here the single cell sequence data for both the MDUF-S3 as well as the FASKD1 in retinal ganglion cells to compare their expression in these different types of ganglion cells, and also to TXNRD2, which is another mitochondrial protein that's involved in glaucoma. Now, the second example that I want to show is CAV2. So one of the first loci for primary open-angle glaucoma was an association with SNPs that are located in a non-coding region between CAV1 and CAV2, which are sort of arrayed in a head-to-tail fashion on chromosome 7. There's always been a lot of attention to CAV1, but recently CAV2 has also been associated with primary open-angle glaucoma. And we have identified also in our exome study a missense allele in CAV2 that is highly associated with disease. What's interesting is that these CAVs, both CAV1 and CAV2, and also CAV3, are involved in formation of these very specific molecular outpouchings in membranes. And there's a number of different biological processes that occur at these membrane pouchings, including regulation of NOS3 and intracellular cholesterol trafficking. So involvement of these 2 caveolins argues that this is an important mechanism in glaucoma and one that warrants further study. And then the last one I want to show you today is also getting at a little bit of something that Dr. Heise talked about as well, and that is this TEC pathway with the ligands angiopoietin-1 and angiopoietin-2. We know that mutations in TEC and mutations in angiopoietin-1 causes severe early onset glaucoma, like congenital glaucoma, in some cases, in some families with variable expressivity. And this is due to the fact that losing the function of this pathway leads to abnormal development of Schlemm's canal. So Schlemm's canal doesn't form, fluid can't get out of the eye, and the pressure goes up. So what we found a few years ago is that there's also an interesting SNP in angiopoietin-2. So angiopoietin-2 had not yet been implicated in glaucoma, and we found that this SNP in angiopoietin-2 is protective. And we also found, using data from the UK Biobank, that the protective SNP reduced intraocular pressure. Now, what's interesting about this SNP is that it lands in a microRNA binding site. And so this change from the G to T at this SNP could actually disrupt binding of the microRNA to the 3' untranslated region of angiopoietin-2, which would then increase expression of angiopoietin-2 because binding of the microRNAs can depress expression. And in fact, what we found is that the protective allele is associated with increased expression of angiopoietin-2 and decreased intraocular pressure. But the really interesting study was one that was just published this past fall in collaboration with Susan Quagen, our collaborator. She made a mouse that actually has this same mutation in it and showed, in fact, that the level of angiopoietin-2 is higher in the mouse tissue and that the Schlemm's canal is larger and better formed in the mouse that has this variant. So this points to this kind of important TEC signaling strength where we have TEC mutations that cause early onset disease as well as adult onset disease, which is interesting in terms of thinking about the full spectrum of glaucoma disease in some of these genes. So just to summarize, genetic studies are identifying hundreds of genes related to pathogenesis and disease risk. Protein function suggests novel disease mechanisms. I can't emphasize enough that the functional studies are very important to show that, in fact, these mutations are really having an impact in defining the mechanisms, and that these mechanisms are important if we're going to think about how gene-based therapies could be used for these diseases. I'm just listing here all the people who are in the International Glaucoma Genetics Consortium because that's a huge source of the data for these experiments, as well as people who collaborate with us on these different projects. Thank you.
And we're open to questions.
In addition to the complex one, 45 subunits told some of the new genes, especially that is involved in the mitochondria fusion and fission. So some of the genes are very, very important. Did you have come across any genes associated with the mitochondrial dynamics?
Yes, actually, there is a gene involved in fusion and fission that is also associated with glaucoma. Yeah. In a study that I did, where it was very simple, I just took all the genes that are in mitocardia, which is for the mitochondria, and then all the genes that were significant in gene-based testing from this most recent GWAS and just looked at their overlap. And we found 16 genes that overlap between glaucoma and the mitocardia. So in another talk, I would talk about all of those.
Thank you.
But the end of the end of S3 is a really interesting gene because we know that it does have it's already a cause of Leigh syndrome if you have 2 copies. So here we're talking about a change in expression that then predisposes to glaucoma.
Great, Dr. Jenny. My question is somewhat related. Were there corresponding mutations in the complex one in the same patient who also had mutation in the nuclear gene?
That also have what?
Corresponding mutation in the nuclear gene that actually codes for mutation.
Oh, well, those studies came from different data sets, so you wouldn't be able to look at the same variation.
So I have a question, Jenny. So
Yeah.
it's very interesting work. So you showed 2 genes of mitochondrial nature, and you said that is with the ganglionic cell layer, right?
The what?
In the ganglionic cell layer?
Yes, right, yep.
So does it have any relationship with the IOP,
With myopia?
with intraocular pressure?
Oh, yeah. So an interesting thing is that in the GWAS, the one before the one that's in preparation, there were 5 different genes in that GWAS that were associated with intraocular pressure that were mitochondria genes. So, and we know that one gene that's a mitochondria gene that's now been in multiple GWASs, TXNRD2, that's involved in NAD metabolism. And we know that gene is expressed in the anterior segment and in the posterior segment. So, it might be one gene from the mitochondria side that impacts both ganglion cell health and intraocular pressure. Yeah, certainly there's a role for mitochondria in the anterior segment, yeah.
Thanks, Jenny. Now let me introduce our next speaker, Dr. Parisamy Samjas, and he's going to talk about translational genomics of eye disease. Samjas, please.
Thank you. Hello. Can you put the slide, please? Okay, good afternoon. So I'm going to discuss with you another 8 to 10 minutes the translational genomics of eye diseases. So this is, for the student's point of view, these are the current trends in genomics, especially single-cell RNA sequencing and the optogenetics like that. So this is the current trend. Concerned with India, there are 10,000 human genome database launched by Ministry of Science and Technology very recently. So this is the newspaper information. Now we have the 10,000 human genome data is ready from 83 population. Because of the heterogeneity from Kashmir to the Kanyakumari is a huge country, and then there's emphasized only 83 populations. Even though we have got the data from 10,000, this data set will serve as a critical resource for genomic research, contributing to the advancement in the personalized healthcare and medicine. So this is the personalized medicine. So if it is a single treatment, it is the serious adverse effect, but the efficacy is low. Concern, once you know the personalized treatment, obviously the efficacy, good efficacy, and then the serious adverse event is very low. That is the point I would like to emphasize. And then this is the very recent information from National Human Genome Research Institute from US. There are 10 series seminars. If you are really interested, please go through those things, because lack of time, I can't be able to explain in detail. So this is a kind of view, the bold prediction for human genomics by 2030. So what we are supposed to do within 2030. So they emphasize them in 10 points. It means you are supposed to get you better to know your own genome. That is very helpful for the treatment. And then the point number 10 is we are supposed to transfer the research from bench to bedside. At least within the year 2030, some 10 to 12 genes should be treated with the gene therapy. And another point they emphasize in the second one, so you should not focus only on the coding region, exomic region, and then you have to focus on the intronic region. That is very, very important. That's what they are emphasizing in this series. And then this is a very recent report from National Eye Institute. They not yet named the eye disease for this particular thing. They phenotypically, they are well documented, but there is no name for this particular disease. And then they did the genotyping of this particular disease or a particular patient. They come up with the TIMP3. This is a name of the gene. And then again, there is a same kind of mutation is, they identified for other syndrome. And then the problem here is why they not yet name the disease. And then they observed carefully in the whatever the gene, the disease not identified, there is no choroidal neovascularization present there, but in the another one is the new choroidal neovascularization is there. That is the main difference. So it means this one is emphasizing we are supposed to examine each and every patient carefully. Then we are supposed to note the microstructural changes in the patients. So then we can enrich knowledge in the field of ophthalmic genetics. And then this is a very, very interesting paper. It came in Progress in Retina and Eye Research. This is one of the high impact journal, around 29. And here what they are emphasizing, suppose we have the 19,000 human genes, and there is no more, new gene discovery or association studies. Those are very, very limited. If you see even the high impact journal, we can't able to see either the new gene or the association studies. Here, now the researchers, they are transferring the research from bench to bedside using the already known genes, either the retinal genes or glaucoma genes. Here they are emphasizing, consider the disease, it is related to the retina. It may be a mild, moderate, severe, or profound, or end stage. And then now they are adopting the gene editing technology. It may be a gene-specific therapies, or DNA editing, or RNA therapies, or stem cell-based therapies, or the optogenetics. So the green bar represent. Suppose consider the diseases in the early stage, you can adopt a particular augmentation. Even it is in the late stage, there is no very less number of retinal cells. If it's so, you can adopt the optogenetics technique. Optogenetics technique is nothing but even you supposed to express the loads, light sensitive protein in that particular region. If it's so, definitely you can provide the hand movement vision for the patients. That's what they're emphasizing. Now, these are the current genetic and non-genetic treatment strategies is going on right now. And then obviously people are using CRISPR. This is a very recent report from Canada. I think they are treating the patient with the LCA using the CRISPR. They are correcting. I think the child is getting better and then improving the visual acuity. This is a very, very recent report from Canada, LCA. And then now I think people are using the adeno mediated base editing, as I told you, the gene editing. So here, in this particular paper, it is a very, very recent paper from Nature Communication. Here, this particular, long story short, here they are targeting one particular RP gene. The same gene is causing human disease, and then they are implementing in mouse model. They are correcting, and then they are getting a vision better for the mouse. So that's the paper is emphasizing. Another one is very, very interesting. So you're supposed to use adeno-associated virus to carry your transgene. Meaning, suppose you're supposed to make a right virus. Suppose the lentivirus is expressed very well in mouse, but there are so many, 6 or 7 types are there in adeno-associated virus. Here, interestingly, this is, I think I'm well impressed about their strategy in this particular research communication. What they did, they have taken the capsid region in that particular viral genome, and then they made 17 mutants in that particular capsid region, and then they made a separate virus and then infected. And suppose if you make one particular variation in that particular in the virus, if you use that virus, it is expressed not only in mouse or monkey or rabbit. It is expressed very well in all the human, non-human primates. That is the story. So if it's so, you can take this kind of viruses in future, and then you can put your transgene, and then you can use it for gene therapy or gene editing. And then I think I don't want to go in detail. This is the adeno-associated virus description. Obviously, it is a very 4.7 kb, and then it is a low immunogenicity. People are using this kind of viruses now. And this is the USA approved gene therapies, and especially Luxturna, they are using for LCA. And concern with the injection, the injection, how you are delivering the virus is very, very important. Suppose whatever the paper I mentioned in the previous slides in Nature Communication, what they're telling, they're comparing in mouse, rabbit, and the non-human primate. So they're putting in two injections, and then they're comparing. Once, they're putting subretinally. So what is the effect? And then suprachoroidal. And right now they're telling the suprachoroidal injection is better than the subretinal injection. That is the conclusion. So, and then there are a lot of clinical trials are going on. So we extensively worked, for the past 25 years, you take from cornea to the optic now, we extensively worked. We have our own database.
based on our interest, I think I'm listed out here. So what are all the LCA? If it is LCA, what are all the gene therapies going on in clinical trial? And then concern with the retinitis pigmentosa, as I mentioned, the strategies, I think they're adopting so many strategies to cure RP. And then Leber hereditary optic neuropathy is, again, it's a mitochondrial disorder. Is already in Pascal and Palmer, they are already a lot of research work is going on and clinical trial is going on for Leber hereditary optic neuropathy. And we have a lot of cohorts. And then down the road, if the gene therapy is possible in India, I think we have the patients, they will get benefited through the gene editing technology. And then X-linked retinoschisis, again, it is a very, very rare disease. Concerned with our hospital, even if it is a rare disease, it's not a problem. We can able to collect a huge number of samples. And one of my PhD students recently submitted the thesis, and she got the best appreciation award for this particular publication in IJO 2024. Again, the gene therapy is possible for RS1. So here, again, it is a dry AMD, the same strategy people are adopting. Age-related macular degeneration, cell therapy, drug therapy is going on right now. And for your kind information, for the past 25 years, you name the disease from cataract, POIG, anything, So these are our data, and then we deal with them in so many, genes. And then now we have the data, and then the spectrum of genetic variation in Indian population, we have the data. And then very recently, for the last 4 years back, we got a huge grant from Department of Biotechnology. It's the Center of Excellence for Pediatric Eye Diseases. Now, we are in that particular project, we focused on the Leber congenital amaurosis, Leber hereditary optic neuropathy, juvenile X-linked retinoschisis, and retinoblastoma. And then we are making our own panel. The thing is, we are excluding we are targeting the specific gene or marker is responsible for our population. We have made a panel, and then the frequency of the mutation identification is more than 80 or 85%. If you see the literature, you can able to detect only the retinal thing, only 60%. But our case, we have more than, detection rate is more than 85%. We have our own panel, and we have our own MySeq, and we can able to do it, routinely for the retinal patients. And then this is our data. Again, for your kind information, we have more than 200 LCA cases. And then these are it is not a single gene, single disease. There are 29 genes. The LCA already, we have the Luxturna is there. Right now, people are for other diseases based on the CEP290 and like that, that is based on the region. Right now, they are developing the animal model for gene therapy. So now we have this kind of, mutation patients who are having that mutation, we have that particular data. So once if the Indian government or especially the ICMR, if they give an approval to perform gene therapy in our country, definitely we have the patients, we can able to recruit them for gene therapy, especially for LCA and other retinal diseases. Again, this is for the LH1. Again, it is a mitochondrial disease, and here very interestingly, so we extensively studied the entire mitochondrial genome for these LH1 patients. Obviously, they are not, all the patients are not, positive for mitochondria. You can detect the primary mutation, secondary mutation. The negative cases, obviously they are having association with the nuclear genome. Now we are focusing on, so what is the crosstalk between the nuclear genome and mitochondrial genome? Now, I think Dr. Jenny she mentioned about the glaucoma like that. So now each and every disease, it is having an association with the mitochondria. That's what we are focusing on. What is the specific mitochondria or nuclear genome associated with the LH1? So like that, we can be able to increase the frequency of the detection rate. The problem in India, suppose if we take in LH1 for cases in abroad, 95% of the LH1 patients positive for primary mutation. In our case, it is a very, very low frequency. We can't able to detect more than 30% or 40%. That's what we are adapting. What is the crosstalk between the mitochondrial genome and nuclear genome? And for your kind information, the mitochondria is 16.5 kb, only 13 polypeptides. only 13 genes. The 13 genes having interaction with the nuclear genome. So that's what we are focusing on, but this particular thing. And here again, it is a RS1. Again, it is a gene therapy. People are going on. We have some cases, 22 cases. And then, so ultimately, I think lack of time. So I will come to a conclusion. What I would like to emphasize here, concerned with our India, using the 10,000 Indian genome database, provide a fresh insight to link between the genes and health. I know definitely it will help for the personalized medicine. And we have a lot of patients, a lot of diseases through that, through the gene editing technology, we can transfer our research from bench to bedside. Thank you. Yes, sir.
Hi, I'm David Mackey.
Yes, sir.
A great talk. I love all the Leber's optic neuropathy stuff. A question with your unexplained optic atrophies. Are they actually multi-individuals being affected within the pedigree, and is it consistent with a matrilineal inheritance, or are they just sporadic individuals who could be phenocopies with some other cause?
Yeah. So I think Dr. Mahesh Kumar is our clinical investigator. So we are making into two groups. I think maybe I can able to explain in clinically. So like primary open LH1 is a positive, clinically positive, and then suspect. So we are getting those two study subjects for mitochondrial mutation. So when we screen, I think less than 45% of them are positive for primary mutations. So then we are if it is a suspect case, we are extracting the data, and then we are focusing on the secondary mutations or the negative cases. Obviously we are targeting for the whole exome sequencing. The whole exome sequencing, we come up with some a bunch of genes that may be correlated with especially the complex one and then the mitochondrial dynamics. That is our observation. Very recently we published that paper too.
Right, because this is years ago when we used familial cases requiring multi-generation people affected, 95% of them were explained.
Okay.
But when we looked at our general, does this person have possibly Leber's optic neuropathy, and it's a valid test to do, only about 10% of those people turned out to have one of the main mutations.
Okay.
A flip side of all of this is a surprising finding that was found in the UK Biobank. We've now got it in 4 Australian cohorts, is that 1 in 1,000 of the general population of European ancestry are carrying one of the two main Leber's optic neuropathy mutations.
Okay.
And as the mitochondrial SNPs have been added to most of the arrays, this is now coming up in a lot of clinical testing where a person being investigated for a retinal dystrophy is getting told, oh, you've got one of the Leber's optic neuropathy mutations, but doesn't make any sense with the clinical presentation. And in fact, when I presented this a year ago in the United States, there are half a dozen genetic ophthalmologists who had this happen to them, and we've had it happen twice in Australia. And when we look at the individuals within the UK Biobank, it's on specific low penetrance mitochondrial backgrounds that this is occurring. Are you seeing these surprise diagnoses of Leber's optic neuropathy mutations in your retinal dystrophy screening?
Maybe I will look into the data and then I can,
Sorry, because time's up, we all have to close session. And thank again for everyone join us today. And thank you, everyone.
Thank you.
So all the speakers will go out and take a photograph. The speakers will go out and take a photograph.